I'm Angela Gruber. I work at Prevention Genetics. I'm a geneticist there. And as Michael stated, I'm one of the geneticists that reviews a lot of the RYR-1 cases that we see. So I just wanted to go through disclosure. Obviously, I am an employee at a clinical DNA testing laboratory. I also just wanted to mention that all of the testing performed at our lab is ordered by a qualified healthcare professional. And just meaning that we don't offer direct-to-patient testing or counseling to patients. And we view this as, hopefully you're getting counseled and you're working with a neuromuscular specialist to really have the correct and appropriate counseling, and that those results are explained appropriately when you receive them back. And really the best resource, and I think this has been reiterated by Kim and Livia, for understanding your genetic testing result should go through a genetic counselor, a medical geneticist, or again, a neuromuscular specialist. And I've included a link up here to the National Society of Genetic Counselors. They have a link on there where you can actually put in and find a local genetic counselor, and it's broken down by specialty as well. So I think that's a really great resource for the community here. So I wanted to go through, why have genetic testing? I hope by a lot of the morning talks that have been given already, it's becoming hopefully clear that the genetic testing really can influence a lot of things. The first is it can end that genetic or the diagnostic odyssey, right? So a lot of families go through a lot of different workups and tests, and I think the average time for a genetic diagnosis is stated to be about 7 years. We've seen families that it's been 20 or 30 years that they've had something going on. And so getting that final confirmation can really be a great thing for the family. They can find a home like the RYR-1 Foundation. And there's a lot of resources and such that can be then provided. Another thing is it can enhance genetic counseling. So this has been discussed a little bit, in terms of family planning. Your reproductive risk can be discussed. There's prenatal testing, pre-implantation diagnosis. I believe Livia is giving a talk tomorrow in greater detail with some of those. Another thing is just preventing harm from genetic conditions. And I think malignant hyperthermia is a perfect example of why having that genetic test can be so important, so that, if you go in for a surgery, the anesthesiologist knows what they should be giving you for a drug. Again, you've kind of heard that it can also influence the treatment and management. Currently for RYR-1 right now, there is not a treatment. But I will say that most clinical trials that are going on right now are required that there is a genetic diagnosis or confirmation. The other piece, and it kind of gets into the treatment and management, is, it's personalizing medicine. So really what's happening is that specific drugs are being designed and developed for specific classes of mutations. And a great example of this is a drug called ivacaftor for cystic fibrosis. And what this drug does is it works with specific mutations to increase the flow of chloride ions through that channel. And why I think that's a great example to bring up to this group is, the ryanodine receptor, it's calcium instead. It's a little bit different, but same kind of concept. And then I just also wanted to reiterate that there's the Genetic Information Non-Discrimination Act of 2008. This is referred to as GINA. And so this protects individuals from any kind of genetic discrimination in health insurance and employment. So as Kim just mentioned, I really wanted to start off, RYR-1 genetics is quite complicated. Even me as a trained geneticist, sometimes I see a variant and I don't know what it means. That's just the unfortunate part with genetics right now. We're at a stage where we're starting to learn a lot more. But as has been stated, we can have both recessive and dominant cases. Family members with the same mutation display this variable symptoms or maybe no symptoms at all, depending upon what age they're at. The type of the mutation can also really influence the severity of the disease. So not all changes or spelling changes within the RYR-1 gene are going to be the same. And then there's this other issue of some mutations cause only malignant hyperthermia. Some may only cause myopathy, and then some of them can cause both. And so that's getting at that genotype-phenotype correlation that a lot of groups are really working on to try and understand. So I wanted to show you what does an RYR-1 mutation actually look like. So what's shown here is a sequence chromatogram. And so what you're seeing here for each of these peaks, they actually correspond to a nucleotide within the RYR1 gene. And I also wanted to bring up this point that I know everyone really likes to say mutation. In the clinical genetics field, we say variant. They mean the same thing, but a mutation is simply a change in the DNA. A mutation does not mean that it causes disease. You could have a change in the DNA, a mutation, that is also benign. So I just wanted to clarify that a little bit, because a lot of times on the genetic test results that you may receive, they'll be called a variant, not a mutation. And yeah, just kind of getting at again that change can be something that causes disease or that does not. And this is actually from in the RYR1 gene, and one of these is pathogenic and one of these is benign, and I'm actually going to go through how I would distinguish which is which later on here. So first I want to start with an analogy, and this is kind of funny to me because I told Livia when there was a break that she had presented something very similar, but I like to think of it as that all of the DNA within your nucleus here is a book. And this is going to be kind of getting at that genetics is just a different language. So on the next level, that book is then broken down into chapters, which are your chromosomes within there. And within the chapters are the sentences, and that's actually the genes that are within that DNA, or just the sections of that DNA. And then from there, that sentence is broken down into letters. Again, the language of genetics is just A, T, C, and G. And then the next level that I think is maybe sometimes lost is it's really the protein. So I like to think that if you read a sentence, you're picturing something in your head or your mind of what that sentence means, and that's really that protein that's important, and in this case, it's the ryanodine receptor. So if the picture changes in your head, and this kind of gets at where she had the visual representative of the hat and the bat, then it means that there's probably something wrong, and that's actually a pathogenic mutation that's affecting the protein. So what is the consequence of a faulty protein? Here I'm just showing some of the different structural proteins in a muscle cell. I'm not showing the ryanodine receptor, but more or less if there would be any kind of change within any of these different proteins, what can result is a faulty protein or protein that's not working well. So here I'm just showing a muscle biopsy image of healthy proteins, good proteins, they're working correctly. And this is what a muscle biopsy could look like in more of a dystrophic patient. But I just really wanted to highlight that visualization of the difference if you have a faulty protein. Okay, so here I have, I don't know if it'll be a fun activity or not, but we're going to say that the cat is the color black. We totally did not plan this with the cats, but cat's the theme here. So this is our sentence, right? The sentence is the gene. So now I want everyone to close your eyes, and I'm going to read through some other sentences, and we're going to distinguish if it's a pathogenic or a benign thing. Okay, so close your eyes and read a sentence. The cat is the color black. Okay, now you can open your eyes. So this is something, that picture in your head didn't change, right? So that's kind of indicating there's no difference in that sentence or that gene. You're getting the same picture in your head. All right, now close your eyes again. The cat is the color black. Okay, open your eyes up. Now, if you're from Britain or Canada, you may say this is the correct spelling of color. But so again, that sentence, that didn't change the picture in your head. This is something that, it's a spelling change that would be considered benign. All right, so now change, or you can close your eyes again. Two more here. The rat is the color black. Okay, so did that picture change in your head? And you can open your eyes up. So you should have changed from a cat to a rat, and I'm considering this something that really is changing that picture. So that's something that we would consider pathogenic, because it's a spelling change that's completely changing what that protein is then. And the last one here is the cat is the color gray. Does the color of the cat matter or not? I don't know. So this is where it becomes a variant of uncertain significance in that sentence, because we don't know if that gray color really indicates anything or not. Okay, so how we classify variants in the clinical genetic testing world, everyone uses the same guidelines and rules. So these are put out by the American College of Medical Genetics and Genomics, also referred to as ACMG. So this is a whole long document that has a lot of different criteria that we're all required to follow when we're doing interpretation. And I just want to kind of bring it down to more or less the criteria or evidence is broken down into very strong, moderate, or supporting. And depending upon how many pieces of that evidence there are, will then influence what the interpretation we give is. So the variant categories that ACMG has put out are benign, likely benign, variant of uncertain significance, likely pathogenic, and pathogenic. So, you know, the benign and likely benign variants, these are things that are spelling changes within the DNA that shouldn't be causing a phenotype or any kind of disease. The likely pathogenic and pathogenic, these are things that there is enough evidence to support that they are something that is causing disease. And then there's the unfortunate category, right, of if it doesn't fit into the benign, likely benign, maybe there's not quite enough evidence yet to call it likely path or pathogenic, it gets put into this variant of uncertain significance. So getting into a little bit more of what do we actually use to classify those variants, one piece, and I think Livia had already mentioned this a little bit, is that type of variant that actually is seen. So certain variants, for example, small deletions, duplications, that result in no protein actually being made, they're more likely to cause disease, and so that's considered a stronger piece of evidence then in terms of the interpretation. The other thing we use is how common is the variant? Is it found in a lot of individuals in the general population? If it's a variant that's found in 10% of the population, we don't have 10% of the population walking around with an RYR-1 related disorder, right? So it should be something that's very rare in population databases. Another piece is, has it been reported or published on? So, you know, I think it was Kim that had mentioned, we look through and we see, are there previously reported patients that have that same variant? Are they presenting similar to the patient that we're testing? And then are there experiments that tell us, is the protein faulty? And so that can also be really helpful in the interpretation piece. And the last piece that we'll use are computer prediction programs. However, we have a little not hesitation, but it's just one piece, and it can result in overinterpretation if these programs are not well understood. They only have about 65 to maybe 80% accuracy, so just because a prediction program says this change is going to be deleterious, that doesn't mean for sure that it's something that would be disease-causing. So in terms of what we use for figuring out how common is the variant, there's a database called the Genome Aggregation Database. I've listed the link here. So this is a publicly available database that anyone has access to. And it more or less has the genetic information from about 140,000 individuals. So here I'm showing population frequencies, just fancy name for how common is it in the population. And we see here that this variant overall is at 10% in the different populations here. And so this would be something that's too common in a population to be causing disease. This would be considered a benign variant because it's in so many people, so many healthy individuals in this database. Getting at the question of, has it been reported before? So in my work, we use this database called Human Gene Mutation Database. And more or less what this database is I can put in my favorite gene, and it will list off all variants and the publications related to that particular variant. So here I've just highlighted this 7523 G to A variant, and if I click on that, it will then populate and bring up, okay, yep, here are the PubMed publications. So now I can go through and really look at those publications carefully and assess, is there enough information to say that this is a pathogenic or benign variant? Another piece of if it has been reported previously is the ClinVar database. Again, this is a publicly available database that anyone would be able to go into. And there are interpretations for variants listed in here. However, I will state you need to be very cautious. You don't believe everything on the internet. It's kind of the same thing if you would go into ClinVar. Some of them are very good and they are correct, but there are some that are not correct in there as well. So that's another place that we can use to figure out if something may be reported previously or not. Okay, so now we'll go through. I'm showing that sequence chromatogram that I had shown you previously, and you all are going to help me figure out which variant is pathogenic and which variant is benign. Okay, so first we start with variant number 1. How common is it? And so if we look at the numbers and the information in NOMAD, we see that it's only in one allele of about 250,000 alleles there. That's very rare. And so it's 0.004% in the human population. This means that it could be something that is disease-causing because it's in such a small amount of people. The other piece that we would look at then is it reported in patients? And so for this example, there are several pieces of literature here showing that it has been reported in patients. And if we look at ClinVar, we see that it's also been reported as pathogenic in ClinVar. So is this benign or pathogenic? Pathogenic. Okay, so that's a very general way that we go through and kind of look at a variant. And then I also do have, so this is an interpretation from a patient that we tested in 2017 that did have this nucleotide change. And I'm going to go through later in some slides and really dissect out the interpretation and the pieces that are in there. But I just kind of wanted to go through, you know, we usually say this has been reported multiple times to be causative for central core and malignant hyperthermia. Some of these reports found the variant to occur de novo. 2 other substitutions, so 2 different changes at that same amino acid have also been reported. And that we see that this variant has also been reported by the European Malignant Hyperthermia Group as pathogenic in their database. So there's a lot of pieces of evidence to support that this actually is a disease-causing variant in this individual. Okay, so now we're going to jump back to variant number 2. And so here I'm showing the population information for variant number 2. And this is in 10% of the population. I kind of already probably gave it away that that's too common of a variant in the general public to be something that would be disease-causing. If we go and look to see if variant number 2 has been reported in patients in my HGMD database, we see that it's not there. And then when we go and look in ClinVar, we do see that other labs have called it benign. So is variant number 2 pathogenic or benign? It's benign. So now I want to go through kind of dissecting out a genetic report. This has a lot of text and wording, so I kind of tried to highlight and underline the important things, right? So the first is, how common is it? So we say this variant has been observed in only 2 out of 277,000 alleles in a large population database, indicating it's very rare in the general population. That's one piece of evidence to suggest it could be something that's pathogenic. The next piece is, has it been reported before? This variant has been reported in 6 cases of recessive myopathy. And so we list the different publications then showing that this variant has been reported and segregated within those families. The next piece that we would look at or that we include is the prediction programs. And so in this case, we use 3 different prediction programs, and they all do predict that this could be a damaging change. So all of those pieces together are what help me then to determine and have the criteria to say, yes, this variant is pathogenic for a recessive myopathy. However, it's uncertain for malignant hyperthermia. And that's where I think some of the confusion and complexities of RYR-1 testing come in, because you may have that it's a pathogenic variant for the one phenotype. However, it's uncertain if it could be causing the malignant hyperthermia. Okay, so why is something called a variant of uncertain significance? I just kind of wanted to go through that because I think that's probably where a lot and most of the confusion comes in for patients, families, physicians, everyone involved. So in this case, so this variant we found in a patient, 11,321C to T. It's not been reported in patients, but it's also not very common in the population. So it could be a rare benign variant in this patient, or it could be that it is actually something disease-causing. The change is predicted by prediction programs to be deleterious and result in a bad protein. Again, that's just one piece, and because it's not been previously reported, we just don't know if that actually is true, if it's having a deleterious effect on the protein. So as I'd stated, this could be a rare benign variant, or it could be the pathogenic variant in this individual. There's unfortunately just not enough information at this time for us to make that call. And I did want to highlight that as more people are sequenced and we gain greater genetic information, it'll help hopefully clarify some of these variants of uncertain significance. So I wanted to go through another variant here that's a variant of uncertain significance. And in this case, again, it has not been reported in allele frequency databases, so it's not found in healthy individuals, but it hasn't, again, been reported in the literature. The piece that, we still have to call this a variant of uncertain significance. However, different changes at this same amino acid have been reported. That makes me very suspicious. This is a very suspicious variant of uncertain significance. So sometimes you can have a variant of uncertain significance that maybe leans one way or the other. Again, there's just not enough criteria for me to bump that up to a likely pathogenic or pathogenic variant. And here I want to highlight the importance of a family study of affected and unaffected individuals to see how that variant may be segregating in the family, because that can support pathogenicity and can help upgrade that interpretation possibly from uncertain to likely path. And I just want to go through kind of a quick example of this. So you guys have seen some pedigrees here already this morning. So if the circle or square is filled in, that indicates that that's an affected individual, whereas if it's not filled in, that's an unaffected individual. So here we're seeing an autosomal dominant pattern of inheritance, and we're going to pretend that the affected child was tested, and this is actually that uncertain variant that I showed on the previous slide. So this child's affected, has this variant of uncertain significance. If you're talking with a genetic counselor or a medical geneticist, their first thing should be that, okay, let's test mom and dad if we're assuming dominant inheritance in this case. So if we test mom and dad and mom has this variant, that then can tell us that this variant of uncertain significance is likely not disease-causing, right? So mom is not affected with the same disorder that child and dad are, so that likely means that that's not actually what's causing disease there. Now, what if dad has the variant? So, dad is presumably affected with the same disorder here, and if he has the variant, that's showing that that variant is segregating within the family. And if we take that one step further, and if we were to test dad and his sister and brother and this other cousin, and all of those affected individuals have this uncertain variant, that really provides a lot of strong genetic evidence that that likely is a pathogenic variant then. So I hope that makes sense and kind of highlights the importance of testing through families to figure out these uncertain variants. So with that, I just want to conclude that RYR-1 is a very complicated gene, even for someone that is trained in genetics, and there's still a lot that we don't know. Unfortunately, genetic testing doesn't always result in this easy negative, positive answer. There's a lot of gray zone in between there that you can get back as well. And again, I really just want to emphasize that understanding your genetic testing result is best done through a genetic counselor, a medical geneticist, or a neuromuscular specialist. And here I've just listed some resources I think that have been kind of brought up in some of the earlier presentations as well. So Genetics Home Reference, again, this is a really great resource for general genetics information. Again, here's the link to the National Society of Genetic Counselors. I've included the direct link to the Find a Genetic Counselor. If you want to read more about the GINA Act, I've listed that. And obviously, you should all be familiar, hopefully, with the great RYR-1 Foundation website as well. And then the Malignant Hyperthermia Association of the United States is listed here. So with that, I'm happy to take questions, and hopefully I've helped to dissect out those genetic results a little bit. So thank you.